Hello guys, in the previous video we had seen the importance of cell membrane and cell wall, particularly in the case of plant cells. The second important component of cell is the nucleus. So let's begin from where we had left. The nucleus has a double layered or covering called the nuclear membrane. The nuclear membrane has pores which allows the transfer of material from inside the nucleus to its outside, that is, the cytoplasm. The nucleus contains chromosomes, which are visible as rod-shaped structures only when the cell is about to divide. Chromosomes contains information for inheritance of features from parents to next generation in the form of DNA, which is the abbreviation for deoxyribonucleic acid. Chromosomes are composed of DNA and protein. DNA molecules contain the information necessary for constructing and organizing cells. Functional segments of DNA are called the genes. In a cell which is not dividing, this DNA is present as a part of chromatin material. Chromatin material is visible as entangled mass of thread-like structures. Whenever the cell is about to divide, the chromatin material gets organized into chromosomes. The nucleus plays a central role in cellular reproduction, which is the process by which a single cell divides and forms two new cells. It also plays a crucial part, along with the environment, in determining the way the cell will develop and what form it will exhibit at maturity, by directing the chemical activities of the cell. In some organisms, like bacteria, the nuclear region of the cell may be poorly defined due to the absence of nuclear membrane. Such an undefined nuclear region containing only nucleic acids is called the nucleoid. Such organisms whose cells lack a nuclear membrane are called the prokaryotes. The pro means primitive or primary and karyot means nucleus. Organisms with cells having a nuclear membrane are called eukaryotes. The figure shown here shows a typical prokaryotic cell. It has a cell wall, a plasma membrane, some ribosomes, and a nucleoid in place of a nucleus. The other main component of a cell is the cytoplasm. The cytoplasm is the fluid content inside the plasma membrane. It also contains many specialized cell organelles. Each of these organelles perform a specific function for the cell. Cell organelles are enclosed by membranes. In prokaryotes, beside the absence of defined nuclear region, the membrane-bound cell organelles are also absent. On the other hand, the eukaryotic cell have nuclear membrane as well as membrane-enclosed organelles. Viruses lack any membranes and hence do not show characteristics of life until they enter a living body and use its cell machinery to multiply. This shows the importance of membrane-enclosed organelles. Now let's see what these cell organelles are and what are their functions. Every cell has a membrane around it to keep its own contents separate from the external environment. Large and complex cells, including the cells from multicellular organisms, need a lot of chemical activities to support their complicated structure and function. To keep these activities of different kinds separate from each other, these cells use membrane-bound little structures, also called the organelles, within themselves. This is one of the features of the eukaryotic cell that distinguishes them from the prokaryotic cell. Some of these organelles are visible only with an electron microscope. Some of these cell organelles are the endoplasmic reticulum, the Golgi apparatus, lysosomes, mitochondria, plastids, and vacuoles. They are important because they carry out some very crucial functions in cells. So let's now see what these cell organelles are and their functions. The endoplasmic reticulum. The endoplasmic reticulum, or the capital ER, is a large network of membrane-bound tubes and sheets. It looks like long tubules and round or oblong bags, which are called the vesicles. The ER membrane is similar in structure to the plasma membrane. There are two types of ER, the rough endoplasmic reticulum or the RER and the smooth endoplasmic reticulum or SER. The RER looks rough under a microscope because it has particles which are called the ribosomes attached to its surface. The ribosomes, which are present in all active cells, are the sites of protein manufacture. The manufactured proteins are then sent to various places in the cell depending on need using the endoplasmic reticulum. The SER helps in the manufacture of fat molecules or lipids important for cell function. Some of these proteins and lipids help in building the cell membrane. This process is known as the membrane biogenesis. Some other proteins and lipids function as enzymes and hormones. Although the ER varies greatly in appearance in different cells, it always forms a network system. Thus, one function of ER is to serve as channels for transport of materials, especially proteins, between various regions of the cytoplasm or between the cytoplasm and the nucleus. The ER also functions as a cytoplasmic framework, providing a surface for some of the biochemical activities of the cell. In the liver cell, for example, of a group of animals called the vertebrates, SER plays a crucial role in deoxyifying many poisons and drugs. This picture of cell shows the presence of smooth endoplasmic reticulum and rough endoplasmic reticulum inside the cell membrane in the cytoplasmic area. Now let's move on to the Golgi apparatus. The Golgi apparatus, first described by Camillo Golgi, consists of a system of membrane-bound vesicles arranged approximately parallel to each other in stacks which are known as the cisterns. These membranes often have connections with the membranes of ER and therefore constitute another portion of a complex cellular membrane system. The material synthesized near the ER is packed and dispatched to various targets inside and outside the cell through the Golgi apparatus. Its functions includes the storage, modification, and packaging of products in vesicles. In some cases, complex sugars may be made up from simple sugars in the Golgi apparatus. The Golgi apparatus is also involved in the formation of lysosomes. So what are lysosomes? Lysosomes are kind of a waste disposal system of the cell. Lysosomes help to keep the cell clean by digesting any foreign material as well as worn out cell organelles. Foreign materials entering the cell such as the bacteria or food as well as the old organelles end up in the lysosomes, which break them into small pieces. Lysosomes are able to do this because they contain powerful digestive enzymes capable of breaking down all organic material. During the disturbance in cellular metabolism, for example, when the cell gets damaged, lysosomes may burst and the enzymes digest their own cell. Therefore, lysosomes are also known as the suicide bags of the cell. Structurally, lysosomes are membrane-bound sacs filled with digestive enzymes. These enzymes are made by the RER or the rough endoplasmic reticulum. The next cell organelle is the mitochondria. Mitochondria are known as the powerhouses of the cell. The energy required for various chemical activities needed for the life is released by mitochondria in the form of ATP or adenosine triphosphate. ATP is also known as the energy currency of the cell. The body uses energy stored in ATP for making new chemical compounds and for mechanical work. Mitochondria have two membrane coverings instead of just one. The outer membrane is very porous while the inner membrane is deeply folded. These folds create a large surface area for ATP generating chemical reactions. Mitochondria are strange organelles in some sense that they have their own DNA and ribosomes. Therefore, mitochondria are able to make some of their own proteins. The next cell organelle are the plastids. Plastids are present only in plant cells. There are two types of plastids: chromoplasts, which are the color plastids, and leucoplasts, which are white or colorless plastids. Plastids containing the pigment chlorophyll are known as the chloroplasts. Chloroplasts are important for photosynthesis in plants. Chloroplasts also contain various yellow or orange pigments in addition to chlorophyll. Leucoplasts are primarily organelles in which materials such as starch, oils, and protein granules are stored. The internal organization of the plastids consists of numerous membrane layers embedded in a material called the stroma. Plastids are similar to mitochondria in external structure. Like the mitochondria, plastids also have their own DNA and ribosomes. This brings us to the last organelle which we have to study, that are the vacuoles. Vacuoles are storage sacs for solid or liquid contents. Vacuoles are small sized in animal cells, while plant cells have a very large vacuoles. The central vacuole of some plant cells may occupy 50 to 90% of the cell volume. In plant cells, vacuoles are full of cell sap and provide turgidity and rigidity to the cell. Many substances of importance in life of the plant cell are stored in vacuoles. These include amino acids, sugars, various organic acids, and some proteins. In the single-celled organisms like amoeba, the food vacuole contains the food items that the amoeba has consumed. In some unicellular organisms, specialized vacuoles also play important roles in expelling excess water and some wastes from the cell. Hence, each cell thus acquires its structure and ability to function because of the organization of its membrane and organelles in specific way. The cell thus has a basic structural organization. This helps the cells to perform various functions like respiration, obtaining nutrition, and clearing of waste materials or forming new proteins. And hence, the cell is the fundamental structural unit of living organisms, and it is also the basic functional unit of life. With this, I end the chapter on fundamental unit of life. Thank you.